Good morning, this is Kavita Kundra, and today we are on last part of Mscdfsm biochemistry topic, carbohydrate metabolism. It is part 6. I know this was very long topic and it was lengthy, complex, but it is actually easy to understand if you go through the concepts. Next, we move towards the topic mannose metabolism. Mannose is phosphorylated by hexokinase. Hexokinase is an enzyme forming mannose 6-phosphate. This is isomerized by phosphomannose isomerase to form fructose 6-phosphate, which can be incorporated into glycogen chain. Sometimes the monosaccharides can accumulate in the body and cause biochemical defects. This is one common defect is called galactosemia. What exactly is galactosemia? Galactosemia is the inability of conversion of galactose to glucose, resulting in accumulation of galactose in the blood, known as galactosemia. Means galactosemia is when galactose getting accumulated in the blood. The biochemical defect is usually due to the deficiency of enzyme galactose 1 phosphate uridyl transferase. This is the full name of the enzyme. Initially, galactose gets accumulated in the tissues and then in blood. The major organ damaged by the galactose accumulation is liver. Galactose gets reduced to alcohol called galactitol in eye and cause cataract. Regulation of blood glucose level. Optimal range of glucose level is 4.5 to 5.5 mmol per liter. Various levels of regulations are exerted at substrate level, hormonal level, enzymatic level, and at organ level on carbohydrate metabolism, so as to maintain blood glucose level. Events for the balance. In fed state, clearance of blood glucose is mainly by the liver via glucokinase, and glucokinase, which is inducible enzyme, removes most of the blood glucose from circulation after a carbohydrate meal. Also in extrahepatic tissue such as muscle, adipose tissue, etc., the uptake of glucose is favored by the insulin. When we are in the fasting state, glucose released from the liver increase. Means most of the liver, the glucose is releasing from the liver when we fast due to glucagon, and in the muscle by epinephrine. Glucagon also enhances gluconeogenesis from amino acids and lactate. Both hepatic glycogenolysis and glyconeogenesis contribute to hyperglycemic effect of glucagon, and blood glucose level is maintained. The anterior pituitary gland secrete hormone that elevate blood glucose level and therefore antagonize the action of glucose. Two major hormones are growth hormones and ACTH, corticotropin. The adrenal cortex secrete the glucocorticoids that increase gluconeogenesis by increasing hepatic intake of amino acid, accompanied by increased activity of enzyme of gluconeogenesis as transaminase. Glucocorticoid also inhibit utilization of glucose in extrahepatic tissue. The next effect is increasing blood glucose level. Insulin decreases the blood glucose level, while other hormones increases the blood glucose level. If the blood glucose level is high, then it results in diabetes mellitus. Diabetes mellitus, it is a complex metabolic disorder. We all know diabetes mellitus. It is a complex metabolic disorder characterized by absolute or relative deficiency of insulin. If we are having not much insulin, that means it is diabetic mellitus. Or defects in the insulin action. If we have insulin but its action is not correct or it has some defects, this anabolic hormone, which anabolic hormone? Insulin. Insulin hormone exerts action on key glycolytic enzyme, thus leading to conversion of glucose to pyruvate. On the other hand, the insulin suppresses the action of all key gluconeogenic enzyme. With respect to glycogen metabolism, the excess glucose is converted to glycogen by activating glycogen synthase, thus leading to the glycogenesis and inhibiting glycogenolysis. The lack of insulin reverses these reactions, and antagonistic hormones like glucagon, epinephrine, catecholamines, thyroxine, etc. brings about hyperglycemia. So what is electron transport chain? We all know we get energy from ATP, and every process of cell requires energy. This chain deals with the synthesis of ATP. Which chain? Electron transport chain. Mitochondrial electron transport chain. Mitochondria, we all know, is a powerhouse of body. So mitochondria is the houses the electron transport chain, the reactions of oxidative phosphorylation. Hence the mitochondria is called powerhouse of the cell. Metabolism of carbohydrate, lipids, amino acids yields reducing equivalents such as NADH, FMNH2, FADH2 that undergoes reoxidation. These reduced coenzymes donate a pair of electrons to specialized set of electron transport chain. Ultimately, these electrons are transferred to oxygen. As electrons pass through this chain, they lose more of the energy. Part of free energy is captured and stored by the formation of ATP from ADP and inorganic phosphate. This process is called oxidative phosphorylation. Transfer of electrons. Electron transfer reactions are carried out by wide variety of enzymes, and these enzymes are coenzymes NAD+, FMN, FAT, coenzyme Q. Other name is ubiquinone, non-heme iron sulfur center, cytochrome and metal ions such as copper and iron. The mitochondrial electron transport chain consists of 4 enzymes complex embedded in inner mitochondrial membrane, has 2 free electron carriers. Electrons enter the chain from the carrier to carrier arranged in order of increasing the redox potential and finally reducing the oxygen to. Components of electron transport chain. Complex 1, NADH2 coenzyme Q, NADH coenzyme Q reductase. It contains FMN bound in association with NADH dehydrogenase and FES proteins. The 2 electrons from NADH first reduce FMN to FMNH2. The FES proteins are reduced next, and finally coenzyme Q is reduced to ubiquinone form. Complex 2, FADH2 to coenzyme Q. Succinate, fumarate are linked directly to flavoprotein dehydrogenase, which in turn linked to the cytochrome of respiratory chain. Succinate dehydrogenase and iron FES protein makes complex DIETICIANS. 2. Succinate ubiquinone oxidoreductase. The FADH2 component of succinate dehydrogenase reduces the FES center, which in turn reduces coenzyme Q to ubiquinol. Complex 3. Coenzyme Q, cytochrome C, cytochrome reductase. This complex consists of cytochrome B, an FES protein, and cytochrome ubiquinone, cytochrome C reductase. Electrons are transferred one at a time from COQ with inner mitochondrial membrane. Coenzyme Q function is medium between two electron carrier and one electron, that is a complex 3 and 4. Thus enzyme Q links the flavoprotein to cytochrome B, the membrane of cytochrome chain of redox potential. Q exists in oxidized quinine or reduced quinol. Okay, now cytochrome C to O2. The cytochrome C oxidase complex is the last component in the electron transport chain ferrocytochrome C oxygen oxidoreductase. It accepts electrons from cytochrome C and catalyzes 4 electrons reduction of molecular oxygen to water. 4 electrons plus 4 hydrogens plus oxygen gives water. Electron flow from Q through the series of cytochrome in order of increasing redox potential to molecular oxygen. The terminal reducing cytochrome responsible for final combination of reducing equivalents with molecular oxygen has high affinity of oxygen, allowing the respiratory chain to function at maximum rate until tissue become depleted of oxygen. This is only irreversible reactions in the chain and gives direction to whole processes. Electron transport inhibitors. Antibiotics. Chemical drugs are the inhibitors amityl, antimycin A and cyanide. Oxidative phosphorylation. It is the process by which ADP is phosphorylated by pi to ATP in respiratory chain. It is coupled to oxidation. The phosphorylation reaction is associated with complex V, which synthesize ATP utilizing the entry of proton gradient generated by electron transport chain from complex 1, 2, 3, 4. These complexes are also called phosphorylation sites. 2 electrons are required to reduce 1 atom of oxygen to water, and therefore oxidation of 1 molecule of NADH or FADH2 corresponds to synthesis of 3 or 2 ATPs and reduction of oxygen.